Okay, let's go ahead and get started. Hello, everyone, and welcome to another exciting installment of the Computational Toxicology Communities of Practice. My name is Sammy Hamp. I am a Public Affairs Specialist in the Center for Computational Toxicology and Exposure. Today we'll be hearing from Dr. Thomas Knudsen. He is a Developmental Systems Biologist in the Center for Computational Toxicology and Exposure, and he will be talking about researching developmental toxicity models. Before we get started, I just want to make sure everyone can please mute their lines. If you are calling in, you can do that by hitting #6. And then without any further ado, I will hand it over to Dr. Knudsen.
Thank you, Sammy, and thank you for folks for joining in. So October is now being more and more known as Children's Health Month, with the focus on raising children's environmental health awareness. Children's health outcomes result from multiple causes. There's genetics, nutritional, socioeconomic factors involved, and of course prenatal and childhood exposures to environmental factors. Some of the adverse outcomes that the EPA is concerned with are things such as respiratory diseases, childhood cancer, neurodevelopmental disorders, obesity, and adverse birth outcomes, which is what I'll focus on. Adverse birth outcomes.
It looks like your screen share might have dropped off. I'm not seeing any of the slides.
How about now?
There we go. It's all good. Thank you.
You still see it, Sammy?
Yep.
Okay. So adverse birth outcomes include preterm birth, low birth weights, birth defects, infant mortality. The U.S. occurrences of birth defects is about 3% of live births, and for low birth weight, it's about 11%. So assessing developmental toxicity during pregnancy has a critical role in setting health and environmental policy. And teratogenesis is one of the major concerns, teratogenesis being chemically induced abnormal development. It's a major concern in the animal models that have human relevance, such as pregnant rats and pregnant rabbits. The term new approach methodologies is probably things that you're familiar with. I won't get into the details of NAMs here, but for NAMs, for developmental toxicity, what we're looking at trying to do is shift developmental hazard detection from animal models to in vitro data, in silico computer models, and focus on quantitative effects on biological pathways. Embryonic stem cell lines are among the most promising alternatives in NAMs to pregnant animal testing. And I'll be talking today about work that's being done on embryonic stem cell lines at the ORD at EPA. I did want to bring up a disclosure, so there is sensitivity in terms of any research dealing with cells derived from fetuses. All of the work that we do here involving human embryonic stem cell lines is compliant with Executive Order 13505. That ensures that it's ethically responsible, scientifically worthy, and conducted in accordance with applicable law. All of this work is on stem cell lines. Nothing is directly from the embryo. These are all cell lines that are approved and registered in the NIH stem cells database. The cell lines I'll talk about include the human H9 cell line from Y-cell. We have a contract that we had with Stemina Biomarker Discovery that generate a lot of the data that I'll be talking about. There's also additional work that's being done in-house at the EPA using the human cell in the NIH repository, as well as a number of pluripotent stem cell lines that are being used, including several from human systems and the J1 mouse cell line. So with this disclosure slide, I'm basically saying we haven't done anything on embryonic stem cell lines that's not compliant with federal law. Novel features of pluripotent human embryonic stem cell lines. There's really three things that are of importance. Number one, they are self-renewal. Cells proliferate and replicate themselves indefinitely when cultured under the appropriate conditions. Secondly, they can differentiate into multiple cell types. So during self-renewal, they can be grown for several months and still can be induced to form spontaneously or by directed effects most cells of the embryo or the fetus. That's what pluripotency refers to. And third, they have actually a pretty amazing capacity for self-organization into rudimentary tissue structures. We can call that autopoiesis, the ability to self-organize. So self-renewal, multi-pathway differentiation, and self-organization are three things that make it very appealing for potential NAMs for developmental toxicity. Embryologically, human stem cell monolayers can recapitulate the epiblast of the early embryo from day 5 to 7 in the mouse, roughly 2 to 3 weeks in humans. So here is a embryoid body that's spontaneously developed in vitro from stem cells, and here is what the epiblast looks like in a mouse at this stage in development. We have a primitive streak. It's basically a bilayered system. The primitive streak has cells that migrate through and form mesodermal components and endodermal components to give rise to the primary germ layers. So pluripotent embryonic stem cell lines capture many of the key features of embryogenesis that are uniquely covered in some of the guideline prenatal developmental toxicity animal studies, such as OECD Test Guideline 414, in which pregnant dams or does, rabbits usually, are dosed during the period of organogenesis. And so what we're asking this primitive embryonic stem cell type and culture to do is to essentially recapitulate a lot of very complex biology of development that's covered in animal studies. So can it live up to the challenge of NAMs? Well, this is what we're trying to predict from this guy. We're trying to predict what's happening over this period of time and what might be susceptible to chemical disruption in vitro that could be predicted as a developmental hazard in vivo over this period of time. In humans, it would be roughly the 3rd through the 8th week of gestation that covers the organogenesis period. The embryonic stem cell system does not encompass the full complexity of anatomical development that we see over this period of time. It's blind to the precise spatial and temporal control of cell-cell interactions that drive this complex morphogenesis. It can miss developmental effects that are secondary to maternal or to placental toxicity. Placental cells generally are not represented in these cultures unless they're added. And of course, there's a variety of ways that maternal toxicity can affect development. There's also uncertainty in terms of post-organogenesis vulnerability, as well as postnatal manifestations of developmental problems. So what happens when things are occurring later than, say, the 8th week in a human or the 15th day in a mouse or rat? Cross-species extrapolation is important. Most of the work that's been done with embryonic stem cells and toxicity testing has been done in mouse systems, mouse embryonic stem cell lines. And so we need to be able to extrapolate that data to the human situation. And if we're using human embryonic stem cell lines, we obviously want to extrapolate that to animal effects. Stem cells have the capability of limited xenobiotic metabolism, but there's other considerations for internal exposure and dosimetry that need to be considered. So toxicokinetics has to be applied. And finally, uncertainties in translatability of all of this information into an intact embryo, toxicodynamics. So we can get lots of information, then we have to start piecing it together and try to build an integrated model of predictive developmental toxicity. I'm still with you, Sammy?
Yep.
Good. Well, a lot of work has been done using mouse and human embryonic stem cell lines in vitro, not only for toxicity testing, but in general for tissue engineering. A group has been put together to do a detailed literature review of the conceptual and practical considerations that must be given to understand readiness of the stem cell data for regulatory purposes, especially if we're going to be implementing these for NAMs. There's a manuscript that's almost ready to undergo technical clearance. The project was initiated by the Japanese Center for Validation of Alternative Methods. A number of the folks involved are listed below from across the globe. And the things that are being considered from the literature work is things such as what is the chemical domain of applicability, what types of readout endpoints are effective or less effective, what are the standardized protocols that need to be considered, what does interlaboratory reproducibility look like with different stem cell models, different stem cell platforms, what is the biological domain of applicability of stem cells for predictive toxicology, and finally, how do we begin to assess predictive power? What are the best ways to do that using information that's out there? Just one simple slide showing you the extent of information that we've found through this project, looking at the testing data in particular that's available. So we used a number of literature mining tools and curation approaches, and essentially identified about 1,500 PubMed articles that could be boiled down using artificial intelligence to about 333 relevant publications. And what we did was we divided these into two groups. Those publications that have tested 10 or more chemicals as the primary goal for comparisons. And then another, that was 18 publications, and then another 174 publications that tested 1 to 9 papers. So we're using those as evidentiary support. Overall, about 1,250 annotated chemicals have been identified. There's certainly lots more that are out there in the literature that are proprietary chemicals that have been used in pharmaceutical space. We obviously didn't include those unless they were identified by some type of annotation. Across the 18 papers, the most overlapping compounds across those studies, 17 of the 18 papers have looked at all trans-retinoic acid, 16 of them looked at 5-fluorouracil, 14 looked at methotrexate, and these are all strong teratogens in almost all species that have been evaluated. So they serve as a good way of anchoring studies, platforms, et cetera, together. The most common use categories from the pharmaceutical space were anti-infective agents and enzyme inhibitors, and from the chemical space, pesticides. The literature base now includes a paper that we have published earlier this year in Toxicological Sciences on a data set in ToxCast, including about 1,065 different chemical samples using the Stemina biomarker discovery contract vehicle. And that's what I'll talk about now is the results from that work. The assay from Stemina, the commercial assay, is called the DevTox Quick Predict Assay. As I mentioned earlier, we had a contract with Stemina to run their assay on over 1,000 ToxCast chemicals. This assay was developed from Jess Palmer in her work as a graduate student at the University of Wisconsin. They published the platform in Birth Defects Research in 2013. And what they identified was pluripotent H9 embryonic human embryonic stem cell metabolomics assay that identified the potential developmental toxicants in a test set with 77% accuracy. So they identified 57% sensitivity. In other words, they picked up just over half of the positives in their test set, but with 100% specificity overall. The assay you may be familiar with, it's based on a relative change in two metabolites, ornithine and cystine. Ornithine is released by the H9 cells into the culture medium over a 3-day period of the assay. Ornithine is a product of several pathways in the cell, including the urea cycle, polyamine and pyrimidine synthesis, and probably has a very strong connection to mitochondrial biology. Cystine is present in the culture medium at the start of the culture, and so it's brought into the cell, and it's used, it enters the glutathione synthesis pathway, and it's involved in redox cycling. What Jess Palmer found in their metabolomics profiling is that the relative change in the amount of ornithine released to cystine utilized by cells over that 3-day period, when that ratio drops to a critical level, which they identified as 0.88, it's normally somewhere around 1.1, that indicated the potential for teratogenicity or developmental toxicity in general. So the teratogenic index, as was originally defined, is the concentration of a chemical or drug that caused a drop in the ornithine to cystine ratio to a critical level of 0.88. Well, we've, as I indicated, published that we've now run that platform with over 1,000 chemicals in ToxCast, using both screening at single concentration and follow-up in definitive concentration response. The targeted biomarker is given the teratogenic index, again, is the ornithine to cystine ratio. And that's measured with a counterpart of viable cell number in the cultures. Methotrexate is used by Stemina as a plate level standard to calibrate the system at different concentrations. It's also one of our ToxCast chemicals, and in fact it behaved in ToxCast just as it does during their plate level calibrations with respect to the concentration that would give a predicted teratogenic index, which in the case of methotrexate was somewhere around 59 nanomolar. In this study, we tested, as I said, about 1,065 different chemicals. The data has been pipelined into our in vitro DB version 3 database, which is what holds the ToxCast Tox21 data. And currently now there's well over 1,000 different assay features in that database. So Stemina data has now been added to that. We call this the ToxCast_Stemina data set. And it's now available at EPA's CompTox Chemicals dashboard. The dashboard actually has all of the data that we've collected, and Stemina provided a lot of quality control data. So if you're interested in accessing this database from the chemicals dashboard, I would first urge you strongly to read the paper and understand all of the different quality control metrics that may not be so important in using the teratogenic index from this study. Overall, we identified about a 19% positivity rate. So in other words, about 1 out of 5 chemicals we tested gave an indication of a potential teratogenic hazard. And of course, this is a hazard prediction. It's not a risk prediction. You'd have to obviously determine if the exposure is relevant to assess the risk. But what the database will give is the concentration of each chemical that would be predicted to hit that teratogenic index. I'll just show a couple of examples. One of the things, if you're familiar with the Stemina data set, the Stemina assay itself, you'll realize that our profiles don't look the way that the commercial presentation of the assay is reported. We have purposely graphed out all of the data in the same way we graphed all of the other ToxCast assays. So if you're familiar with ToxCast, you'll recognize these profiles. So the targeted biomarker, which is in the blue box, in this case it's comparing all trans-retinoic acid, which turns out to be the most potent of the chemicals we tested, with a teratogenic index predicted at 3 nanomolar, versus retinol, which is inactive at the concentrations that were tested here. And that retinol, of course, is the precursor to all trans-retinoic acid. Retinol is vitamin A, which is taken into the pregnant mother, converted to retinoic acid either in the liver or in the embryo. And all trans-retinoic acid is a morphogenetic compound in the embryo. So retinoid signaling pathways are very important for a number of normal and toxicological effects during early organogenesis and differentiation. Here's another example of a pesticide, Lozafop P-butyl, which is an active herbicide. It's present in 2 stereoisomer forms, the S form and the R form. The P-butyl is the active form, it's the active herbicide. And both compounds are present in ToxCast. The active herbicide was active on the stem cells. The inactive form was not active at the concentrations tested. I've listed also the developmental lowest effect levels observed for these 2 compounds in a rat and rabbit. And just real quickly, if you'll notice, both of them had developmental toxicity when tested in pregnant animals. So it's possible that in vivo one form is converted to the other. The inactive form may be converted to the active form in vivo, but the stem cells are able to distinguish the 2 forms and make a prediction as which one is likely to be a bigger developmental hazard. But then, of course, you have to look at considerations such as in vivo metabolism and what's happening to the inactive form when it's given to a pregnant mouse or a pregnant rat that's not happening when it's exposed in the culture model. Here's another example for a parent compound, Benamel, and its presumed proximate toxicant, carbapenem. In this case, this stem cell assay was able to pick up activity in both cases, both the parent compound and the metabolite, at roughly the same order of magnitude. And there's a number of cases of false negatives, so things that are not detected in this data set that should have been. So diethylstilbestrol, of course, is an important developmental toxicant. It was not detected in this platform. And in fact, most estrogenic compounds were not detected in this platform. And cyclopamine, which is a powerful animal teratogen. Not quite sure if it's a human teratogen or not, but it was not picked up. We tested it several times. The contractor at Stamina says they will pick it up, but at high concentrations. So here's another case of a false negative. And finally, here's an example of 2 clinical compounds that were under development as potential angiogenesis inhibitors for use in halting tumor growth. One is a synthetic thalidomide analog, 5-HPP33, and the other is a synthetic fumagillin analog, TNP470. Both of these compounds were predicted by the assay to be developmentally toxic, but at quite different levels. 5-HPP33, its teratogenic index was around 11 micromolar, whereas TNP470 had a teratogenic index predicted at 17 nanomolar. And we didn't have any rat or rabbit data to compare. these compounds too in terms of their potential in vivo toxicity. However, Ed Carney's group, while he was alive, Ed Carney's group at Dow Chemicals was interested in testing these two compounds in their whole embryo culture system, rat whole embryo culture system. So the predictions, again, for both of these compounds were made from the human stem cell work. And they did rat embryo culture, which they're experts at, over a 48-hour period, and exposed the rat embryos to either compound at a variety of concentrations. For 5-HPP33, they observed an AC50 on embryo viability in culture at 21.2 micromolar. For TMP470, they observed an AC50 at 0.04 micromolar. So although that was dysmorphogenesis. So although the phenotypes differed between these two compounds and their effects on the embryo, both were confirmed to be active, and in fact, the AC50s that were observed in culture were just above the teratogenic index predicted by the data set, the stem cell data. So in fact, the stem cell biomarker really did predict a teratogenic threshold that was validated, at least in this case, using rat embryo culture models. I am not aware of any in vivo studies that have been done with these chemicals to know what would happen in terms of a fetal outcome, so I can't indicate what the ultimate prediction would be. But of course, vascular disruption is one of the major mechanisms by which a number of pharmaceutical compounds are known to cause abnormal development. So from this work, we can ask, does that biomarker predict something new? So getting more mechanistically into what's going on. So the biomarker had applicability for predicting the effect. Can we learn something new from that prediction? So from that same experiment, we took the embryos that were exposed to that threshold level and concentration just above and below, and used RNA-seq profiling to examine how the exposed embryos reacted to the threshold concentrations of either compound. And this was measured after only 4 hours exposure in the embryo culture model in Ed Carney's group. The data was processed by Kate Saili and Jill Franzosa at the EPA here. And what they were able to identify was a number of unique pathways affected by each chemical, but in common between them, the unique effect involved spliceosome RNA metabolism and pathways for proteasome ubiquitination. And this is interesting. One of our former postdocs who went on to Celgene did some beautiful work that they just published on thalidomide effects on human induced pluripotent stem cell lines, looking at mesodermal and endodermal differentiation. And what they found was that, as expected from what is thought to be the target for thalidomide, cereblin, that's a protein involved in proteasomes. And thalidomide's teratogenicity in the stem cell model that Blair et al. Published was attributed to proteasomal degradation of the SAL4 protein in stem cells that were induced into a limb-bud mesodermal lineage. So here's an interesting and very independent connection between thalidomide pathway, thalidomide analogs, thalidomide-induced vascular disruption that may involve proteasomal effects. So it's sort of learning something new from basically a new application of existing knowledge. Okay, so if we then go to the next step and ask the question, how do we determine the effectiveness of the prediction from the human embryonic stem cell biomarker? So within our ToxCast data set, we have 42 compounds that are fairly well curated developmental tox reference compounds. and these compounds are often used to validate alternative platforms for developmental toxicity testing. Across the 42, we identified a number that were true positives, a number that were true negatives, and then a few that were false negatives. We did not detect any false positives. The overall balanced accuracy of this comparison was 82%. We had picked up about 2/3 of the positives with perfect specificity overall. And those performance measures are exactly consistent with the original pharma-train model published by Jess Palmer. Now, in fairness, some of the compounds are the same compounds that they originally trained the model with, but keep in mind that this was completely independent through our ToxCast plates that were sent to them, blinded, and they did the work not knowing anything about the compounds that we provided them with. So 82% is pretty good. Some folks tout getting up to 90% accuracy with this or with other stem cell platforms. But many of the assays that have undergone validation exercises have been tested with really only a limited set of very data-rich chemicals. And so it's often true that when those data-rich compounds are expanded to other broader landscapes of chemicals, they don't see that very high predictive capacity. And in fact, some of the particularly sets that are trained with or evaluating pharmaceutical compounds, which have excellent data from in vivo effects, it appears that the performance of those assays are actually inflating the predictive capacity of that assay against a broader space of industrial chemicals or commercial products. And this situation overall in the field has hampered regulatory acceptance of individual alternative assays. So in this example, I talked about 42 well-curated reference compounds, but we have, of course, many more that we can correlate within our ToxCast ToxRef databases. And in fact, across the 1,000 chemicals we tested in this assay, we have animal data in the ToxRef database on 432 of them. And I'm just going to walk through this very simple looking slide, which I think is very informative. We're working from the right to the left. On the x-axis, the 42 chemicals I just mentioned are shown there. That's actually the peak performance that we see, which is about 82% balanced accuracy. Now, if we start digging into our ToxCast chemicals and look for things that give very well-curated effects in pregnant rats and rabbits in particular, we can expand that 42 set to 127 sets of chemicals that are concordant in their responses, both positive and negative, between studies in a pregnant rat and a pregnant rabbit. So the two species positivity or negativity would be a good indication of human potential developmental hazard, at least in the pharmaceutical world. If we go further, we can add another 160 or so chemicals that may not be quite this, as concordant. So in these chemicals, the developmental lowest effect level on a fetus was below the maternal effect level for maternal toxicity, but this didn't have to be concordant between the rat and the rabbit, so it could have been either the rat or the rabbit. As you can see, the balanced accuracy in the red line is dropping because the sensitivity of the assay is dropping. We're picking up fewer of the positives. If we go down and go to the whole 432 compound set where we have animal data, well, we've taken all of our very confident calls out for the higher stringency models, the higher curated models. And now what we've added back in is largely chemicals where the DLEL may be low, but it is consistent with the maternal LEL. So we don't know if this is having an effect directly on the fetus, or is it because the mother is being affected. And if we look at just any study at all, whether it's prenatal developmental study or not, so now we can expand the set to 1,040 chemicals. And so as you look at this plot, you'll notice that as you gain less confidence in the output, the endpoints, the accuracy of the prediction drops down to 50%, which is a coin toss. And that's primarily because the sensitivity is lost. The animal studies are picking up positives that the stem cell assay is definitely not picking up. But as we get more tight in our developmental endpoints, the stem cell assay does a lot better in terms of its performance, in terms of balanced accuracy and picking up the positives. So essentially the take home from this slide is that predictivity of the human embryonic stem cell biomarker declines as the fetal outcomes gain less concordance between rat and rabbit studies and also begin to show more concurrent maternal toxicity. And that was the main conclusion from our publication. Well, so not so fast, as our rat friend is telling us. Don't throw it out yet. There's ways of dealing with the implications of weak sensitivity. So in any in vitro assay in a NAM, the predictive power of an assay is going to depend on its biological relevance as well as a standard against which the prediction is validated. With 1,000 in vitro assays in ToxCast, we can begin to use machine learning and mechanistic modeling approaches to better characterize the biological domain of applicability. In our publication, we actually did this looking at 337 different biochemical assays in ToxCast in the NovaScreen data set. What we found was that if we look at the true positive space and look at which chemicals hit that were true positives and hit assays in NovaScreen that we could map to pathways, the flow of regulatory information to the AKT FOXO signaling pathway turned out to be the strongest predictor on the positive side. In terms of false negatives, many G protein coupled signaling pathways working through calcium signaling, as well as steroid hormone signaling pathways, were not sensitive in this assay. So they're sort of the poster child for the insensitive domain. So what I want to focus on is the positive predictivity in this model, which is driven in our hypothesis to FOXO. Now I'm going to get more into speculation. We haven't done the definitive studies, but be happy to work with anybody that would like to work with us on this. But we think that FOXO is important, and it takes us again to the proteasome ubiquitination pathway shown down here on the lower left. So Todd Zerlinden, who did the biochemical profiling with the NovaScreen data, said, okay, let's try this again, but let's use all additional ToxCast assays. He did a number of things. We've got a manuscript in preparation, so I won't go through the details, but what we did was we divided the 432 chemicals with animal data into two groups. One group of very well curated effects that he uses a training set, 183 compounds. And the rest of the chemicals we held out for testing a training model. Todd worked through a dozen or so different machine learning approaches. He used some unsupervised feature reduction approaches, and he used Bayesian methods to shrink the assay space down to the most significant assays based on maximum prediction and minimal residual error. And it turned out the same machine learning algorithm was the same in both cases. It gave the best prediction and it gave the least residual error. He used that logistic regression approach with Bayesian regularization to mine the data set. And what we could do is pull out 7, actually more than 7, but the most significant model was 7 additional assays besides stamina that were given the best prediction, the best performance against that training set. It turns out that overall, the 1,000+ assays, the stamina data set was the strongest predictor of prenatal developmental toxicity in the whole system. And that's shown in the 90% credible interval off to the right here. The next best predictor was XBP1. XBP1, in this case, is an antigen assay that's showing activation in the unfolded protein response that independently targets FOXO1 for proteasomal degradation. So here again is the connection to proteasome. And there were a couple of other assays that picked up things such as cyclopamine is missed by stamina assay, but it was picked up in other assays. Some of the estrogenic and androgenic compounds that are missed by the stem cell assay are picked up as well. And the strongest negative predictor we had was the TSPO, mitochondrial 18K translocator. And that's now known from a new study to be pharmacologically connected to the XBP1. So one can ask the question, does this integrated statistical model add to the stamina prediction on its own? The top I've already shown, that was from the graph I showed earlier. Just going to focus on the color coding of the balanced accuracy. The darker the blue, the better the prediction. It's sort of gauged against the medium standard here. When we did the training model, we got very good results. We got almost up to 78% balanced accuracy. But when we applied that to the held out test sets, it barely gave us a prediction above coin toss. But what we noticed was that if we start digging deeper into that test set, about half of those chemicals we could actually do a better job predicting, and particularly in rat, when we get down to a third of them or even a tenth of them, we can essentially reach with a subset of compounds the level of confidence that we can get in the training model. But again, that's only a subset of the compounds. We don't know why these are performing better than some of the others. It could be maternal tox, it could be a number of features. We're looking more into these types of things. But we also noticed that the rat prediction was a little bit better than the rabbit predictions. So there may be some differences between maternal effects in rat and rabbit, or dynamics between these two species that are important. And we don't know yet. We're looking into that. Sid Hunter's got a mouse cell assay where he's tested the stem cell predictions, and he's looked also across the ToxCast library, and at least 7 of the 8 in our augmented model he's picking up as well. Kate Saeli did some work with the human embryonic stem cell line that was induced to it was an induced pluripotent stem cell line that was induced to endodermal lineage. Cog Closure's group had published a few years back that SOC17 could predict a large pharmaceutical set of developmental toxicants with 87 to 94% accuracy. And we know that retinoic acid, which was our most potent compound, is an endogenous signal in mesendodermal patterning. So we asked the question, what would a toxicological tipping point look like for retinoic acid on stem cell differentiation? And I won't go through this. We did that work with Imran Shah's group and published that last year. But the bottom line was that the tipping point that was identified using an RNA-seq approach reflects an imbalance in genes regulated by EOMES, the eomesodermal gene, which is a T-box family member that drives endoderm specification and mesodermal delamination during gastrulation. It's also very important in driving memory cell, T-cell differentiation. And as it turns out, EOMES is another connection to the FOXO pathway. At least that's been shown in memory T cells. So again, we have another connection to the hypothesis that potentially proteasomal function can explain positivity in this stem cell assay. We have the study from the NovaScreen work. We have the study from Todd's expanded augmented model. And we have the work from Kate Saeli's work on stem cell differentiation. So that really leads us to the hypothesis that stem cell positivity is linked to altered homeostatic control of the FOXO1 axis as a negative regulator of differentiation. And again, that needs to be proven. Chad Deisenroth and John Gamble are working with a similar approach now to take that endomesodermal lineage and expand that across to get a broader representation of germ cell differentiation and looking for tipping points at the molecular level. And this work is just really getting started. I just wanted to call that work out. Okay, so just to start wrapping up, in terms of computational NAMs for predictive developmental toxicity, I showed that high-throughput data-driven models can confidently predict developmental tox potential, at least based on extrapolating to pregnant animal study designs. But there are limitations, of course, as I've indicated, in the biology of the system or in the metabolism in terms of classifying teratogenicity across a mechanistically diverse landscape of compounds. Possibly bringing the embryology into the fold, particularly embryology of chemical tipping points, will further improve our mechanistic understanding and translating NAM data to probabilistic effects and early life stage phenotypes. And I just want to end this talk with two new ways that we can evaluate the embryo in this context and show you why sophisticated computer models are going to be needed in the immediate future. The first example comes from Malte-Spielman, looking at single-cell RNA profiling of early mouse development, organogenesis stages. And in this incredible simulation that's running, this is showing single-cell RNA profiling. Each dot is the transcriptome of an individual cell. These cells have been compiled from embryos between 9th and 13th and a half day of gestation, so it covers a good part of organogenesis. And in doing their RNA-seq, I'll run it one more time. So they can put together the different cell clusters that each individual cell belongs to and look at the state relationships. And what they found was over this period of organogenesis, there's 38 very well curated cell types that develop in the embryo. But they identified 655 different molecular states of those 38 cell types. Many of the cell types are connected together by similarities in states and in transitional cells that go from one state to the next. So single-cell RNA profiling, which was identified by Science Magazine as the breakthrough of the year 2018, is one important approach. And so a key challenge for science and technology is to critically define what the sentinel cell populations are in a perturbed system of the embryo. And how does that chemical injury propagate across the toxicological tipping point, ultimately to manifest as a structural defect or altered developmental function. And the second example, which comes from our own work, is now trying to model individual cell behaviors and their dynamics using agent-based modeling. Here's a simulation of the primitive streak developed by Richard Spencer at our Environmental Modeling and Visualization Lab, where he's simulated now the signaling that's required for migration and lineage specification of mesodermal populations going to the dorsal nerve cord, the somites, the limb buds, and extraembryonic tissues through the primitive streak, which is the hallmark of gastrulation. The bottom line here is that cell position matters. You don't get that in a stem cell system. The cells are positioned stochastically, but in an agent-based model, we can now recover the rules, and essentially what we can do is recode the genomic blueprint of body plan formation. And I just want to leave with the quote that comes from a famous developmental biologist, Lewis Wolpert, a few decades back, where he says, "It is not birth, marriage, or death, but gastrulation, which is truly the most important time in your life." So we can begin to think about recoding the body plan computationally using agent-based models in combination with single-cell RNA-seq data. And I think this is a good ground for community of practice in terms of thinking about new hypotheses and approaches that can be used. So with that, Sammy, I'm just going to thank all the folks involved in the work that was presented. And if there's any questions, I guess we could take them now, or I could take them online.
Great, yeah, and it looks like we already had a question in the chat. I think you can pull up the chat box. You should have like a little Adobe Connect thing where you can look at it, or you can just exit out of your slides, yeah. Okay.
Can you just raise the question, Sammy? I don't see it in particular here.
Yeah, I will go ahead and just read that off. So this one is from Michael Goldsmith, and they want to know, are there any assay-specific issues that could make some of the chemicals problematic in the assay? Sort of assay amenability or chemical compatibility issues for the assay medium and well composition, etc. Also, are the applied doses and the cellular concentration scaling evenly across chemical space, or do some chemicals not deliver intracellularly in vitro as they would if they were in a biological system?
Yeah, Rocky, your question's very relevant. I don't have the answer. It's the same concerns you'd have with any in vitro system. I could tell you that we are working through a Tox 21 program with Nicole Kleinstruer and Annie Lumen, Annie at the FDA and Nicole at the NTP, in terms of bringing in more of the toxicokinetic modeling aspects to this. So it's definitely on the radar. That's a great question, though.
Great, and then it looks like we got another question from Jess Palmer from Stemina, and then they wanted to say, to answer your questions, we have not had any chemicals to date that we have not been able to test, at least at some concentration. The solubility will determine the concentration we're able to evaluate, which will determine the relevance of the results in vivo, i.e. do we test high enough to see an effect? Okay, I would expect that. Okay, cool. So that was more to answer the question in there. It looks like we do have some other people typing though.
Yeah, thank you. And of course, Jess is one of our important collaborators on this work, and she's been incredibly helpful to work with as an employee of Stemina, doing a lot of the work. She was our project manager on the contract.
Gotcha. And so it looks like we have a question from Valerie Adams, and they want to know, is there any overlap with genotoxicity in these sets of chemicals?
Valerie, I would guess so. I don't know for sure. I could tell you that one of the assays in Tox 21 that always seems to correlate with some of our positivity is actually a DNA damage assay. I didn't list it here. We didn't go into a lot of the details with the second tier of predictive assays, but there's a good chance that there could be some genotoxicity involved, but I don't have the answer to that. Would love to talk with you if you know more about it.
Cool. And then it looks like we also got a question from Elizabeth Mendez, and they are asking, on slide 18, you mentioned concordance between dev-rat and dev-rabbit studies. Do you mean any type of developmental effect or the same type of dev effect across both species?
Yes, Liz, it was basically any developmental effect. So is there a DLEL in a rat and is there a DLEL in the rabbit? We would call that concordant. But we did not dig into the specific phenotypes. We are doing that now. We are looking, we have a postdoc, Jossie Piero, that's going to be doing that, looking at more detail in some of the chemical endpoints. She's doing that for the retinoid pathway to get started, but we can apply the stem cell data to look at skeletal defects and phenotypes across the species. Thanks for the question.
Gotcha. And so another one from Richard Becker. They say, I might have missed it if you remove substances that produce developmental effects at doses that overlap with maternal tox, how is the predictivity/balance accuracy impacted?
It's destroyed. What we're finding, Richard, is that when we try to predict the held-out set, there's almost no good negatives in there where you have a negative response between two species, because that would have been put into the training set. So our negatives in that case are largely chemicals that were just tested in one species and not the other. Our positives in all cases were positives where the developmental LEL, lowest effect level, was at or above the maternal LEL. So we cannot rule out that the effects are due to a maternal effect on the fetus. And I think because of that, a lot of the prediction is really trying to predict noisy data. I think the stem cell assay does a good job predicting direct effects on the fetus, but it's probably not in this form predicting toxicity of the mother. I know Stemmen has got a number of versions of the assay that they've developed with different biomarkers for cardiotox, for neurotox. So it may be that looking at some of the other biomarkers in their platform could help sort some of that out. Thanks for the question, though.
Okay, cool. And I'm not seeing anyone typing right now. I'll give everyone like a minute or so just to see if anyone has any other questions. But I just want to cover a little bit of housekeeping for the communities of practice. We're going to have a slight change in the schedule for the next couple months. We're going to have one final webinar early in December, so it's going to be happening on December 3rd, same time, just from 11 to noon. That will be reflected in the invites. We are not going to have separate webinars for both November and December this year. Just wanted to let everyone know about that, and the presentation in December will be about CCTE.
Okay, thanks, Sammy, and thanks, everybody.
All right, well, I think, I think we can call it right here. Just give everyone a couple minutes of their day back. But thanks, everyone, for coming. And thanks a bunch, Tom, for the great talk.
Thanks.